Professor Toftgård, thank you very much for joining us so soon after the prize has been announced. Could we begin by you telling us who has received the 2009 Nobel Prize for Physiology and Medicine?
So the laureates are Elizabeth Blackburn from the University of California, San Francisco. It's Carol Grader, working at Johns Hopkins University School of Medicine, and Jack Szostak from the Harvard Medical School.
And so what have they received the prize for?
So they've been awarded a prize for the discovery of telomeres and the enzyme telomerase and how it protects the chromosomes.
And so for people who haven't heard of telomeres and telomerase before, could you explain it for them in simple terms, please?
So I would say that our genes are encoded by DNA, and the DNA is present in the chromosomes in the cell nucleus. And the telomeres, they are the ends of the chromosomes, and they have an important function to protect the chromosomes and maintain the integrity of the chromosomes.
And so what would happen if they weren't protected, if the chromosomes weren't protected by these protective caps known as telomeres?
So then the chromosomes would get shorter with every cell division, and in the end that would lead to loss of genetic information, but it would also mean that the chromosomes will be degraded, and that will start the degradation process in our cells and eventually lead to cell death.
So it has profound implications for cells and organisms as a whole. What sort of implications for the organism does shortening of telomeres have in terms of normal cell function and disease?
So it's evident that this is a fundamental biological mechanism, and mutations in the telomerase genes have actually been detected in a certain group of inherited diseases. And they are characterized by difficulty to maintain stem cells, particularly in the bone marrow. So if you have too little telomerase activity, the stem cells in the bone marrow are at risk, and some of these patients develop severe anemia.
So we know about this implication in disease, and we know in cell function, but the story in which we discovered the role of telomeres and how telomerase is important in maintaining telomere length is a story that's told in several chapters. And I wonder if we could go through each of those chapters in turn to discover how we went from the beginning to what we know now. And so the story starts before the discoveries were made that were being rewarded with the Nobel Prize in Medicine. And I wonder if you could tell us a bit about what we knew about how DNA is copied in the first place.
So what was known up in the 70s was that DNA is copied by DNA polymerases. And this is a rather complicated process when both strands of a DNA thread has to be copied, and it proceeds smoothly in one direction, but in the other direction it is a stepwise process. And the problem is that when it reaches the end on one of the strands, it's not possible to copy the very end of the chromosome. And scientists didn't know how this could occur and how this could take place, and many different theories were around to explain it, but in fact nobody knew.
So people knew about a mechanism for copying DNA, but what the missing piece was how DNA at the very ends is copied.
Yes, and it was also known that that was very important to keep cell happy and alive.
And so then we reach the first chapter of the telomere and telomerase story. And could you talk us through the first steps and the key experiments that were performed that revealed the existence of telomeres?
Yeah, so in fact it was that Elizabeth Blackburn were studying some certain specific types of chromosomes in a single cell organism, Tetrahymena thermophila, which has these chromosomes, and she discovered and found that they had a specific sequence at the end. Although she was at that time also interested in understanding how these chromosomes were maintained. Now, Jack Szostak, he had a different problem. He was wanting to make artificial mini chromosomes and put them into yeast. And when he made such, he discovered that if they were linear and they could not be maintained in yeast cells, they were rapidly degraded.
So what was happening was that there was a piece of DNA that was being discovered at the end of chromosomes, which was repeated many different times, and this was occurring in an organism that lives in water, which is very distant from human body. And another person, Jack Szostak, was working on yeast, and he was trying to work out why the chromosomes he was introducing into yeast were degrading. So then we reached the next part of the story, which is the key experiment. Could you talk us through that, please?
Yeah, so as in all good science, there are lucky moments, and one of these lucky moments was when Elizabeth Blackburn and Jack Szostak met at a conference in 1980, and they heard then about each other's results, and then they decided to test the possibility that the end sequences from the tetrahymena could actually function in a very different species yeast, which in evolutionary terms is far away from each other.
Yes. And so what happened?
Yeah, so then they made mini-chromosomes just like the one that Jack Szostak had made before, but now they put tetrahymena telomere DNA at both ends, and when they introduced these mini-chromosomes now into yeast cells, now they could be maintained for long periods of time and in an intact manner.
And so it's quite incredible that the experiment worked, because you have two totally different organisms taking a protected cap from one organism and attaching it to another seemed to work fine.
Yes, and I think that was one of the most important aspects of the experiment, because that immediately said that this must be a very strongly conserved and fundamental mechanism that was discovered.
And so the next chapter of the story involves discovering how these telomeres are made in the first place. And could you talk us through that, please?
Yes, so the hypothesis was then that there might be a new and unknown enzyme that was involved in actually adding DNA sequences to the telomere ends. And Carol Grader and Elizabeth Blackburn started a search for the enzyme. And so they used tetrahymena cell extracts, added DNA sequences corresponding to the telomere DNA and building blocks for DNA. And then they could actually analyze if an extension took place. And again, on Christmas Day 1984, Carol Grader could actually detect the first signs of enzyme activity in her extracts. And that was then the starting point to go on.
So it's a delightful Christmas present. But what exactly is the machinery that creates telomeres?
So it turns out that it is a unique enzyme that has a protein component that actually has a catalytic function, and it has an RNA component that is intrinsic to the enzyme that serves as the guide or the template to determine which building blocks that should be added and in which order.
So the RNA acts as the template for construction, and the enzyme that's part of the complex is the machinery that creates the DNA strands.
Yes, these two components are actually sufficient to make the reaction work in vitro. Now later on we know that in real cells there are also many other components that are involved and are surrounding these key components that we are talking about here.
And so we have this lovely finding in different organisms that shows that how the ends of chromosomes are constructed and how it protects the chromosomes from degradation. We then need to move to how this is important to the cells that are in yours and mine bodies. So what was the next leap of knowledge that we had in order to know that this is the case?
Well, since there was early observations that also said that if telomerase activity was compromised, then cells would actually age or senesce in culture. So it was thought that maybe this is a key mechanism to aging. However, now we know that aging at the organismal level is a much more complex process, and there are many factors that influence aging, and telomere length and telomerase is one that is important, but there are many others. Now, the other key thing that also came quite early was the discovery that most cancer cells have very high telomerase activity, and that is needed since cancer cells proliferate very frequently and in one sense can be said to have eternal life. And at the same time, cancer cells also have abnormal telomeres, so that has raised hope that one might be able to develop new therapeutic strategies to treat cancer based on blocking telomerase activity or to eliminate cells that have high telomerase activity. Again, one must be careful also with potential side effects, since it's also now known that stem cells have telomerase activity, and that activity is important for the maintenance of the stem cell compartment.
So where are we with regards to understanding the role of targeting telomerase as a potential therapy?
So early clinical trials are ongoing in the cancer area, primarily with two approaches. The one approach is to block telomerase activity, and the other one is actually to try to teach the immune system to eliminate cells which have high telomerase activity. And I think there is one phase 3 study, but most of the other studies are phase 1 or phase 2, and so we still have to wait until we get conclusive results and know if this will be have efficacy towards cancer or not.
So going back to the discoveries that have been rewarded with the Nobel Prize this year, these were made in the late 1970s and the 1980s. So is there any particular reason why the candidates for the prize were deemed particularly worthy this year?
No, actually, so the first key discovery was made in 1982, the second on enzymatic activity in 1985, and the RNA component in 1989. So that is the time scale. Going through different discoveries and weigh them against others is a lengthy process, and it must also be very evident that this is a general process, and also then the Nobel Committee goes through who actually did the discoveries in great detail. So there is no particular reason that it's this year, but now all the knowledge and all these factors that I mentioned are around, and they were selected to be at the top of the list this year.
And one things that the achievements highlight is the value of curiosity-driven research and the value of high-risk, high-reward research. And that's a message that's always been told by the Nobel Prizes since 1901. Is this a message that the Medicine Committee is keen to promote more than ever?
I think that with the Nobel Prize in Physiology or Medicine, we are very careful to select discoveries which are of fundamental importance in biology, but also have clear implications. But it is the fact that if you discover a new fundamental biological mechanism, it's bound to have also medical implication. And this is the case also with the discovery of telomerase. And another important factor is that also curiosity-driven research, also in organisms that may seem far away from human beings, turn out to be very important. In this case, a unicellular organism, a ciliate, and yeast, but they were enough to reveal such a fundamental biological process that has all the importance also in human beings.
And that's a great message to end on. Professor Toftgård, thank you very much.